First of all, I would like to thank the Royal College of Veterinary Surgeons for inviting me to speak in the Fellows in Focus series of talks. My name is Mandy Peffers. I'm a Wellcome Trust Clinical Intermediate Fellow at the University of Liverpool, and I'm a Fellow of the Royal College of Veterinary Surgeons through a thesis. My talk is entitled From Bovine Embryo Transfer Vet to Wellcome Trust Clinical Intermediate Fellow, and I wanted to tell you about my journey into research as an academic in a university from being an embryo transfer vet. So this slide shows you the timeline of my career to date. So I was a failed vet. I got 2 B's and a C at A level, and so I went to the University of Leeds and did an animal science degree. And from that, I still was interested in being a veterinary surgeon, so I applied to go to the Royal Veterinary College into the second year, but I also applied to do a PhD, and I was lucky to be offered both of those. In the end, I decided to go to the Royal Veterinary College and do my veterinary degree. Following that, I did a reproduction internship at the University of Glasgow with Dr. Mike Harvey, and that gave me some initial research experience within the lab and also in some clinical research. At the end of that, again, I was offered the opportunity to do a PhD, but I felt it wasn't right for me at that time. So for the next 10 years, I became a cattle embryo transfer vet for a cattle breeding company in Cheshire called Genus. In 2007, I became redundant from that post. The company stopped that facility. And so for a year, I worked as a locum vet, both for my husband's practice, of which I was a director. It was an equine and small animal practice, but also for out of hours practice called Vets Now. At that time, I became interested again in potentially following a research career. So I went to meet a colleague who I knew from the Royal Veterinary College, Peter Clegg, at Liverpool University, and asked him what the opportunities were for doing research. And he told me about some ring fence money from the Wellcome Trust to try and encourage vets to get into research. So from 2008, I did a Masters of Philosophy in osteoarthritis with him and Simon Chew at the University of Liverpool, and that was called an entry-level fellowship. Following on from that, Wellcome Trust were offering 6 years of integrated fellowship. So 3 years was for a PhD and 3 years was for clinical training. So I was lucky enough to be awarded one of those, and I undertook my PhD on the proteomic and transcriptomic changes in osteoarthritis and cartilage aging. The second part of that fellowship was supposed to be a residency, but I was still really interested in doing wet lab work. My PhD had given me the tools and the hunger to find out more about my area of research, osteoarthritis. So I managed to persuade the Wellcome Trust to fund me as a postdoc for 3 years. After that, I got another fellowship. Again, I was very lucky to be awarded a clinical intermediate fellowship, and that carried on some of the work from my PhD, looking at small nucleolar RNAs or SNO-RNAs in osteoarthritis. In 2019, I was promoted to personal chair at the university. So, as you can see from my CV, I've undertaken something called career diversification, which is defined as spreading your resources both in terms of time and effort among several opportunities as opposed to maintaining a single career, for example, as a clinical practicing vet. So I've been an embryo transfer vet, I've been a locum vet, I've worked in spay clinics, a animal rescue, I've been a practice vet for small animal and equine, and I've also been a research student, and now I'm currently an academic. So I think this highlights the great varied career that you can have as a veterinary medicine graduate. And I wanted this talk to highlight really to vets out there that you don't have to do the same thing for your whole of your veterinary career. If you've got an interest in research, you can certainly pursue that, which is what I did. And I wanted to give you a few bits of information on the way things that have helped me and things that are important as a researcher generally. So the first thing I wanted to talk about was making the most of your contacts and networks. So this is really important to get a start in research, really. For me, having been graduated for I think about 11, 12, 13 years before I went back to Liverpool to do my MPhil, I wasn't quite sure where to start, so I made the most of a contact that I'd made at the Royal Veterinary College. His name is Professor Peter Clegg, and at the time he was a resident in equine surgery at the RVC. So he gave me the information about the types of funding I could apply for and what I would need to do. And then with his contacts, we managed to develop a project idea for the PhD, and that was trying to stratify osteoarthritis using proteomics and transcriptomics, these new omics technologies that are used routinely now in research. And the Wellcome Trust liked you to undertake some of your work in other universities, and because of my family situation, I had 2 young children at the time, I couldn't move, and a business in North Wales, I couldn't move to the other side of the country or another country to do it. And so Pete used some of his contacts to help write this proposal, and one of them was Dr. David Thornton, who Pete had worked with already at Manchester University, so I spent 6 months at Manchester studying in his group. And the other was a proteomics specialist, Rob Bainan, at the University of Liverpool, and I spent a large part of my PhD doing proteomics with him. So I study osteoarthritis, and I study small non-coding RNAs in osteoarthritis, and I use human, equine and canine tissue and biofluids. And I think being a veterinary surgeon allows you the opportunity to look at diseases holistically, particularly age-related diseases, chronic age-related diseases that affect both our companion animals, sports animals such as horses, and ourselves. And that's what I found really interesting about studying osteoarthritis. So once I'd got my PhD, I started publishing papers from the work that I'd done within my PhD, and I was lucky enough to get quite a number of publications. So the first paper I published was looking at quantifying how proteins change in osteoarthritis in an absolute level. And then I also looked at proteins that were changing in synovial fluid from horses with and without osteoarthritis. I undertook an externship at a specialist lab in Amsterdam, and some work I did there ended up with another chapter to my thesis and a paper, and that was looking at spatial resolution of proteins within cartilage in osteoarthritis and cartilage aging using something called mouldy imaging. And then the final part of my thesis was looking, again, a proteomics approach, looking at how extracellular matrix molecules are cut up by enzymes that we know are important in the degradation of cartilage in osteoarthritis, and that was this neopeptides paper. And towards the end of my PhD, RNA sequencing was becoming more common and more economically viable to do. So I did a transcriptomic or an RNA-seq study on the differences in the transcriptome, so messenger RNA protein coding genes in cartilage aging in the horse, and so that was the final paper. The next thing I wanted to touch upon was opportunities. So the great thing about research is you get to meet loads of interesting people from all over the world, and there's lots of other opportunities that might come your way. And so I'm quite a yes person, and when I was doing my PhD, I took as many opportunities as I could. So an example of that is the Farm Animal Proteomics Cost EU initiative that was out there, and from that I went to a meeting on the farm animal proteomics. I undertook a course on mass spectrometry imaging, on mouldy imaging, and went to a lab, and that's where I undertook the work to look at differences, spatial resolution of proteins that are changing in cartilage and aging and osteoarthritis. Another opportunity I had was through the Newton Researchers Links, and I went to Kazakhstan to a stem cell meeting with other scientists from Kazakhstan, and through that I later got a PhD student from Kazakhstan who came and joined my group. And then the final example here is there's a group of conferences called the Gordon's Research Conferences, and they're specialist conferences in all sorts of areas, and I've been lucky enough to attend a couple of those on cartilage biology. And this image is researchers from throughout the world. The best research was in cartilage biology, and there's a mixture of early careers researchers, PhD students, as I was at the time, and PIs, so people who are heads of research groups. So all those opportunities allowed me to develop collaborations and increase the number of projects that I was working on. So in the second part of my integrated fellowship, I did a postdoc, and so I was lucky enough to get some money from the MRC and versus arthritis to provide bench fees, and the things I was studying within those 3 years was stem cell aging. And so we were looking at how human mesenchymal stem cells change with aging using a multi-omics approach. So we did mass spectrometry imaging, we did label-free quantification proteomics, RNA sequencing and small RNA sequencing, DNA methylation, and that produced the papers on the left-hand side. At the same time, I took some of my data from my PhD, so these were changes in small non-coding RNAs called SNO RNAs, and developed a project looking at SNO RNAs as potential biomarkers of osteoarthritis. And finally, some of the opportunities that I previously had access to, and through collaborations through those opportunities, I got involved with a number of other papers, so a couple of tendon papers, both with Peter Clegg and Siobhan Thorpe and Ethna Comerford, and also some biomarker papers and looking at the effect of an extract of broccoli on osteoarthritis as a potential treatment. So my intermediate fellowship was based partly on work from my PhD. So if you remember, I said one of the papers that I looked at was transcriptomic signatures in cartilage aging. So this was an RNA sequencing study on young and old horse cartilage. And we found changes in these molecules, didn't even know what they were, I had to Google what a SNO RNA was, and that's in the table on the bottom left, and how they were changed. And I presented that work at a World Osteoarthritis conference, a human conference. And scientist Tim Welting on the top of this slide here came and chatted to me, because he was also studying a particular SNO RNA called RMRP RNA, which is mutated in a human disease called cartilage hair hyperplasia that causes a type of dwarfism. So from that meeting and from the opportunity to speak at that conference, I met my main collaborator, and I developed my intermediate fellowship around SNO RNAs. And from that intermediate fellowship, we found that indeed there was a catalogue of SNO RNAs that are differentially expressed, that are changing in both aging and osteoarthritis. And we looked specifically at some of those SNO RNAs, such as U3, and found that it was really important in protein translation within the chondrocyte. And further work following on from that included looking at small non-coding RNAs in equine synovial fluid in early osteoarthritis, and looking at small non-coding RNAs in equine chondrocytes and how they change in aging. And these epigenetic molecules are really important because they change the expression of protein coding genes. And none of this work would have been possible without collaborations, and these are some of my collaborators from both the UK and throughout the world. So Tim Welting, who I've spoken about, Eva Skolibrand from Sweden, who has been a great collaborator within equine biomarker work, Steen Jacobsen, who is an equine surgeon from Copenhagen, who is a current collaborator looking at extracellular vesicles within synovial fluid using a horse osteoarthritis model, Ian Clark from the University of East Anglia, who I undertook the broccoli study with, Karina Wright, who's another biomarker collaborator, and she's looking at biomarkers of responders and non-responders to biological therapies in human osteoarthritis. Victoria James, who's from Nottingham Vet School, who again I met at another conference, and she was talking about EVs. And I didn't even know what an EV was. Again, I chatted to her, developed a collaboration, and now we've got a horse race betting levy board grant looking at EVs in osteoarthritis. Simon Jones from Birmingham, who is studying long non-coding RNAs, a different type of RNAs, and we've got a study at the moment looking at senescence and the role of EVs in senescence. And Siobhan Thorpe from the RVC, who's a tendon biologist. So not only are these collaborations important within career development, but they give you a sense of fulfillment. You meet some really interesting people. Most of these people are much cleverer than I am. They are predominantly basic biologists, and yeah, they've really helped me along the way. I guess another area of consideration when you're doing a research degree is peer support. And I've been lucky enough to be involved in a department in a wider research group called the Comparative Musculoskeletal Biology Group at the University of Liverpool, initially based at the vet school and then later in the middle of Liverpool. And they've provided support in understanding techniques, in basic questions, mental health support, and we really hope we help each other along the way. So having a peer support system, whether that is within your research group or externally, is really important. And finally, but not least, is a family and work-life balance. So I don't think you can have a fulfilled career in veterinary medicine without a supportive family. And mentally, you need a good work-life balance. So initially, when I was an embryo transfer vet, it was not 9 to 5 job, but I knew exactly when my hours were, and there wasn't any out of hours work, and that worked really well when I had young children and when they were babies. So I started my PhD when the youngest, Archie on the top right, was 6, and Hamish, who is now at uni, would have been 10, and so it was a bit easier for me to juggle academia and being a student again. And I couldn't have done that without the support of my husband, who's also a vet, Andy Peffers. So having that mental resilience comes from the people around you, I think, in part. So currently I've got an independent research group at the University of Liverpool. We study epigenetic changes in musculoskeletal diseases, particularly age-related diseases such as osteoarthritis and tendon disease. This is my group before COVID, on bottom right, and we're a mixture of vets, scientists, PhD students, master's students and postdocs. And I'm lucky enough to have funding from the Wellcome Trust, the Horse Race Betting Levy Board, the Horse Trust, Pet Plan and the MRC. So thank you to the Royal College of Veterinary Surgeons for allowing me to speak. If you're interested in a career in research or you just want to chat, my email is there and I'm happy to speak to anyone. Thank you very much.